Hi, my name is Darryl Oliver. I'm a second year medical student at Meharry-Vanderbilt College, and I'm here to present my poster. Genetic variants of the APC7 lipid transporter displaying differences in ATP hydrolysis. This research is important because Alzheimer's disease, I'll call it AD, affects millions of Americans today. The APOE or apolipoE protein 2 is believed to be protective against AD. 3 is about average, and 4 is thought to make people more risk and have greater risk and more susceptible to developing AD and earlier in life. For African Americans, APCA7 is believed to make them more at greater risk for developing AD and earlier in life. And we believe that by 2060, the number of Americans with AD should increase, or may possibly increase, to 60 million. So talking about the APCA7 protein, we transfected some 293S cells. These are human embryonic kidney cells, with them using a green fluorescence protein to be able to detect them under a microscope. And we see here that they, regardless of the variant, whether it's the wild type, 395, 215, or 319 variant, they do localize to the cell membrane, and that helped to reinforce our theory that it is a membrane protein. What we know so far about it is that it is a protein that helps to, as a lipid transporter, functioning to accept the lipid and kind of move in a hinge-like fashion to transport lipids from within the cell to the APOE apolipoproteins on the cell membrane, which also plays a significant role in the clearance of amyloid beta buildup in the brain. And we know that when there's a defect in amyloid beta, when there's a defect in APCA7, we see that there is a decrease in lipid homeostasis, and also an increase in the number of amyloid beta buildup. It exacerbates the amyloid beta deposition and impaired memory. So we know from experimentation that APCA7 helps to regulate the levels of lipid, amyloid beta, and its ability to be cleared by microglia, supporting cells in the brain, and also the phagocytosis and apoptosis of cells by macrophages. So as I mentioned, with our research, we found that APCA7 does localize to the cell membrane, and we also, here we use B-actin as for normalization. And here with these bar graphs, you see that the level of expression was about the same for each one. Over here, we see that there's the wild type, and in this 319 variant of APCA7, both prefer the phosphoinositol ethanolamine and serine over the choline, but we do see a reduced amount of ATP hydrolysis in the ethanolamine and serine variants of the phosphoinositol. And with further research, we'll be able to get a greater p-value and determine how significant this difference is, and if there's a possible link as to why people who have this variant versus the wild type may be more susceptible for developing Alzheimer's disease. And we also see differences in ATP hydrolysis between the control, the APOE, and the APOAI variant of the apolipoprotein. Between the wild type and the 319, we actually see greater ATP hydrolysis in our experiments with the APOAI, and we see reduced rates in the presence of mutations. So that's our work that we've done so far. I'd like to thank our dean, office of our dean, Dr. Benjamin. I'd like to thank the Alpha Omega Alpha, Caroline, King Student Research Fellowship for funding our research, as well as the Alzheimer's Association. And thank you very much, and appreciate you accepting my candidacy. Thank you.